And there's going to be some way for the public to interact and have an input on what they would like to see studied in PKD next. Is that about right?
Exactly.
The premier source of patient-centered information on PKD and how to reverse it. This is Felix Mueller.
Hello everyone, this is Felix, your friendly neighborhood kidney hacker, coming back today with another episode. And this time, I am super happy to welcome Beno Freedman to the show, who is the director of the PKD Invent Center at the University of Washington. And he's a specialist in all things PKD organoids. We met a while back, actually almost, I think it was 10 years ago now, right? I had the honor of visiting your lab and donating some myself and looking a little bit behind the scenes. I saw some of the organoids floating in the jar, which was very exciting. Today, we're going to talk a little bit about that new center that you founded recently and also the newest developments in your research with PKD organoids.
Thanks, Felix. It's great to be here. As you said, we've known each other for now, it's hard to believe, but about a decade. And we've developed, I think, a great relationship, both as colleagues, but also as friends. So happy to be on the podcast.
It's been a long time coming. It's surprising we didn't do this earlier. So
we've spoken about it.
Yes, so we did.
Here we are. This is great. This is great. I mean, I think from our perspective, we want to have scientific dialogue, not just amongst academics who are professionals in this area, but also hackers such as yourself, folks who have a personal interest in it. And it's great that we're doing it.
Yeah. Back in the day when keto wasn't even a thing. So I recently heard you speak actually in Germany, my home country, at the University of Aachen. You were here to talk about some new developments in the area of organoid research. But of course, the listeners who might also have PKD themselves are probably interested to learn like a short version of, okay, what are organoids and how is that better than other research that we're already doing?
Yeah, that's a great question and a good place to start. So to me, an organoid is a collection of cells grown in a dish. So it's grown outside the body that resembles a tissue or an organ of the body. It's one of those things where you know it when you see it. Every organoid is kind of like a mini organ at some level. It resembles, so if you look at a kidney organoid, for instance, which is what we study, there's these tiny tubes that have a very specific architecture and progression of that structure that's unique to the kidney. Mm-hmm. So that's the field. And there are many different ways to grow organoids, but the principal two methods are you either can grow them from a person's adult body, where they have to be derived from a piece of tissue, essentially, but grown from there outside the body, or we grow them from stem cells. And typically these are pluripotent stem cells. So they're very early stem cells, which arise before any of the organs have formed and have this potential to go in all those different directions. So they kind of undergo the process of organogenesis, organ development in the Petri dish. So those are two ways to make organoids. And there's a huge constellation of these organoids out there. They're all a little different. Even amongst the kidney organoids, for instance, you can imagine different labs have different flavors of these or different versions that are all somewhat different, but also somewhat the same. Now, why are they better than other methods? I don't know that they're necessarily better, but they're an alternative, and they give us new information. So traditionally, we've been stuck at two different extremes. We either have animals, which we can study to a degree, and we can perturb to a degree, but they don't really exactly copy human beings, and they're also extremely complex. Or we have cells, which we derive from a person, let's say, but they're pretty simple in the way that they grow in a petri dish. So, they might be human, but they don't have all that complexity, and they really don't show signs of disease. They don't get sick the way tissues or organs get sick.
Yeah.
So an organoid is kind of in between that spectrum, and it gives us a little bit of a window into both the human biology as well as what's happening in between cells and a real well-formed organ.
So when we're talking about making the cells from pluripotent stem cells, you have like a certain method that you use to get them to be a kidney. How does it actually become a kidney and not a liver cell or something else?
Yeah, it's like black magic, basically.
Good.
Every organ lineage has to be discovered, and I was actually one of the first people to figure out how to make these pluripotent stem cells into kidney. Kidney is actually a latecomer to the field, so it only really got discovered in the last decade or so. And it turned out that we just had to treat the cells with a very high dose of a particular chemical, and that essentially jettisoned them off on their way to turning into kidney instead of another lineage. If we used a lower concentration of that same chemical, they would turn into beating clusters of heart cells. Okay, so the conditions were, you can use the same exact conditions, just a gradient of that chemical, and on one side you'll get the heart, on the other side you'll get the kidney.
Wow, that's fascinating. Does that chemical mimic anything that's in the body, or is it unrelated?
It activates a signaling pathway that's in the body, probably multiple signaling pathways. But it would have been difficult to predict that particular pathway was the key ingredient. So it's more been a matter of essentially trial and error over the years of discovering these cocktails that we use to, create the particular organoids of interest.
Right. Okay. So a very, very simple model for listeners might be the Petri dish, sort of 2D, and the organoid is sort of the 3D version of it. We have more dimensions that we can look at.
And many different cell types as well. So the organoids are complex. They have many different cell types in the same culture.
Oh, more complex than a Petri dish could ever be.
Well, the Petri dish, typically the 2D culture is going to, regress to one type of cell, or it'll be a simpler culture. All the cells, they may even all derive from a single cell. A single cell can give rise to many, many clones. So some simple cell cultures are made that way. Whereas the organoids, they tend to be mixtures of at least two different types of cells, but often as many as, 10 or 20.
Okay. And I guess in the actual kidney, it's even a lot more than that.
Yeah, tissues are more like organoids. They've got a spectrum of different cell types present.
But even more than 10, it's probably 100. Or
Right. Yeah, there's more in a sense. I mean, when you get to start sort of deciding what exactly is one cell type versus another, It gets a little bit complicated. So what happens in the body actually, I think, is that there are many defined cell types, but they're well defined. Mm-hmm. Whereas in the organoids, you're right, you have fewer cell types, so the entire spectrum is more limited, but there's also, they're not as well defined because they're outside the body. So they start to lose their identity.
Yeah. It feels a little bit like use it or lose it. So, if you don't train a lot, then you're going to lose muscle. And maybe if there's not a lot of external stimuli, then sort of the cells lose their differentiation, something like that.
Yeah. I mean, I think this is one of the big challenges, right, is because also for maturing the structures to the point where they're able to really function, they need to be tasked with a job.
Right.
And if they don't have that job in front of them, then they get a little confused and can slip into other states and try to turn into other things.
Yeah.
Nevertheless,
that makes sense.
We're not looking for perfection here. We just want structures that we can study and utilize. And for that, the organoids have been great.
Yeah. So I checked out one of your earlier papers and compared it with what you said in the recent presentation. And I noticed a difference. So in the 2017 paper, you discovered that when you don't have any extracellular matrix for the cells to adhere to, and that increases cyst growth. So in Aachen, it sounded like that was the other way around. So when you added extracellular matrix, then cyst growth was reduced.
So I think I need to explain this a little bit better.
Please do. Please do.
The finding that we published in 2017 is that we essentially released the organoids from their adherent cultures into a suspension culture. In other words, the organoids start off, they're growing on a petri dish, similar to any other traditional 2D culture. But they kind of mound up, and they form these structures that grow multicellular layers above the surface of the dish. Now, those, if they have PKD mutations, they will begin to show signs of disease. In other words, they will swell up and form cysts, but it's only a minority of those structures on the tissue plate that do that. So it's about 15, 20%.
Yeah.
It's still very clearly PKD, because if you look at the same cultures without those mutations, they don't form the cysts. So it's clearly specific to PKD. It is PKD in a dish, but it's not every little organoid that forms cysts. Now, if you take those structures and with a little needle, micro dissect them off the plate and pop them into a floating culture, so they're in suspension now, they're floating around with a lot of degrees of freedom. Now, the PKD organoids, almost every single one of them will go on to form a cyst.
Right.
So by toggling that environment, by changing them from essentially a 2D to a free-floating culture, we could dramatically increase the formation of cysts. Now, that's still true, but one thing that we've learned over time about this system is that the cysts are inside out.
Yeah.
Relative to how they form in the body. So the cysts have their tops floating, facing the outside of the cyst. Oh, that's pretty cool. See, sometimes I can get this firework effect to happen. It's sort of it's a superpower of mine. So they're forming outside. They're forming inside out. And in the body, the cells face into the cyst.
And you can see that because of the staining, right? So you have this image, and the outside is sort of red, and the inside is sort of stained green, so you can tell.
Yeah, you can look at features of the cell. The cell has a top and a bottom, and you can stain for those. So that's been a little bit of a challenge for us, because ultimately, it's a head scratcher. It's interesting. But ultimately, we also want to have the cysts be able to form right side in. So what I presented in Aachen is that, and this is still not published, but it's interesting, and we've done it a number of times. If we grow the cells and if we grow the organoids and submerge them in extracellular matrix, then they will form the cysts. They still go on to form the PKD cysts, but they form the cysts right side in.
Yeah.
So providing that cue, that extracellular. So what is extracellular matrix? Extracellular matrix is essentially a gel. It's the stuff between cells. Okay, it's kind of the glue that holds tissues together.
Right.
And it's not normally added to these cells when we're growing them in the lab. So we have to add it in. And if we do that, we can get the cyst to form right side in. I wouldn't say, are they more than if we don't have the extracellular matrix? That we still don't know in that adherent culture condition.
Mm-hmm. That flip in behavior that you observed there, it reminded me of something else that flips sometimes in the Petri dish. I've seen that there's different reactions in conditioned versus unconditioned media. Is that something that you've observed?
Well, conditioned media can be a lot of different things, but typically it means something where there's been cells exposed to the media.
I guess there's something that more resembles the environment that has some signaling molecules that would be in the body versus a Petri dish, the longer you condition it.
Right. So cells, sometimes you have a conditioned media where the cells have been pumping stuff out, secreting things into the media. These can be signals, they can be growth factors, what have you. So we haven't done a ton looking at conditioned media. There were some early studies back in the old days of PKD where, people theorized that there was a secreted factor that could actually be contributing to cyst formation. And no doubt there's some things that are contributing, but I think the question is there anything really critical there?
Is there one thing that's always needed? That's the question that you're referring to.
And it's kind of a chicken and egg scenario in the Petri dish because, for example, our organoids are making their own factors. They're secreting stuff. And then those things that they're secreting are actually, then they're feeling those things. They're encountering those same things that they're secreting, right? So they're kind of swimming in this pool of their own making. Mm-hmm. We don't really see any of that, but it's also impossible for us to fully remove those signals from the system.
Yeah, for sure. And probably having more signals would at least mimic the body more, even though it's not as surgical, right? So it might make it more natural. I saw that there's a couple studies showing that, or at least one that shows that the reaction to LPS, which we have talked about in the past, is flipped. So it increases respiration in unconditioned media, but it decreases respiration in conditioned media. So what you discovered also, or what you presented on, you probably knew it for a long time, is that there's a involvement of the myosin.
Well, it's a pathway. Myosin is a biological pathway, really.
Because in your presentation, it looks a little bit like a muscle that sort of surrounds the cyst itself. You could think of it that way, even if it's not true, because that muscle can sort of ease and have less tension. And you discovered something very interesting around that.
Yeah, this goes back to this study where we had popped the cysts off the plate. We found that they would swell up, and that told us that there was something that they would swell up with a higher frequency, and that told us there was something about the way the cell interacts with its environment that was affecting the penetrance of the PKD, right? And then we started thinking about what are the different types of pathways, what are the different types of ways that cells interact with their surroundings. And we picked a bunch of different chemicals, and these included extracellular matrix and things that affect the focal adhesions, which attach the cells to the surface, and myosin, which is a contractile protein that's involved in the movement of cells. Not just muscle cells, it's most famous for muscle, but there's also myosins in all the different cells of our body, non-muscle myosins as they're called.
Okay.
And what we discovered in a nutshell was that myosin, of all of these pathways, seemed to be the one that had the biggest effect on polycystic kidney disease cyst formation. If we blocked myosin, if we reduced contractility, the cysts got bigger. If we activate myosin and we make things more contractile, the cysts got smaller. So this is now leading to the idea that maybe if we can activate myosin in these cysts specifically through a pharmaceutical approach, then we might be able to develop that as a therapeutic for PKD that's, essentially causing the cysts to retain their shape. And fundamentally, if you think about this, like you were saying, if you think about a tube versus a cyst, a cyst can kind of be seen as a tube that's let itself go, right?
Yes.
That's expanded out. And a contractile protein, a protein that gives cells its shape, like myosin, that could explain how that's actually happening.
So now the question, of course, is chicken or egg, right? So have you had an opportunity to find out, does this increase the pressure inside cysts because it's still trying to be more fluid? Is it still excreting more fluid even though myosin is sort of holding it in? And then follow-up question, do you know, because I don't, if this mechanism is involved in cysts in general, so cystic acne, for example, or is it a PKD mutation specific thing?
Oh, those are good questions. I think we don't know yet. I think there's a couple ways this could be working, right? It could be that the cells are weaker and the pressure inside them stays the same, right?
Yeah.
As a normal situation, in which case the cells just can't resist it as much and slowly expand. Or it could be the cells are equally strong as the normal cells. There's just a difference in the pressure somehow. Now, I favor the first explanation because the pressure, to my mind, in the organoids, there isn't a ton of pressure. So it's hard for me to understand how pressure could be building up inside those organoids at that level.
You don't think it's like secretion itself? There's like secreting,
Well,
then
you know,
it sort of has to give way.
Secretion would tend to go from the direction of the cyst, from the direction of the cells out. So in the organoid cysts, which face outwards, that would actually be pumping into the surrounding media, into the surrounding liquid. So it wouldn't be pumping into the cyst.
You mean when it's flipped?
Yeah. The organoids are flipped because they're flipped in their, they grow that way. The cysts in the organoids, they don't flip at any point as far as we can tell. They form naturally inside out relative to how they would form the body. So I feel like that makes it unlikely that it's a pressure buildup situation. But I never say never as a scientist.
Yeah.
I think we really need to look carefully and understand what's going on. In terms of your second question, so I favor the explanation the cells are weaker at some level. They've got reduced myosin, they've got reduced contractility, and the good news is there are compounds, there are, investigational drugs and things like that out there that could perhaps make them stronger and counteract that weakness. Of course, all of that needs to be studied and proven to be safe before any of it is tried in people, which is what we're trying to do.
Yeah.
Now, the second question was whether it goes beyond polycystic kidney disease. Is it a general thing that could apply to other sort of cystic disorders? Well, what I'll say is that, whatever is happening in PKD certainly affects other organs of the body besides the kidney, right? So we know that cysts form, for example, in the liver, to some degree in the pancreas. There's also some aneurysms that can form within the blood vessels.
Yeah, sure.
So even PKD, it's clear that there's a sort of general cyst pathway. And then there's a spectrum of related disorders to PKD, such as polycystic liver disease that have similar types of cysts but work with different genes. So there's some kind of central pathway there. Now, does it get to the point of being blisters on the skin? I don't know. These are the types of things that we'll just have to see as we go along. Our focus is on PKD.
Yeah, for sure. I guess the reason that I ask is because I just can't really imagine a cyst anywhere in the body being able to expand that way, wherever it is, without the mutation, with activated myosin. If we're going with the model that's like sort of a muscle that's tensing the cyst up, keeping it from expanding, that would also be present in any other type of cyst without PKD mutation.
Right. There's always going to be some balance in any cyst structure. It's kind of like a balloon. There's a pressure inside that balloon, and there's an external wall that's resisting that force and applying an equal and opposite force. Yeah, so that is definitely a general truth. We can learn a lot from looking at those types of physical systems, about PKD. It does seem to me to be some sort of almost a pressure blockage. And there's been a lot of thought about that over the years.
Yeah. So when you were saying you think of the PKD cells as just a little bit weaker, that totally rings true because we know that tight junctions are affected and the e-coherence seem to be somehow worse off than without the mutation. Is there a connection between just having like a less strong or like a more permeable epithelial layer and this myosin connection?
I don't know yet. I think we don't quite know what the properties are of the cysts as they're forming. We know that after they form, they change a lot. Certainly as the cyst gets old and gets big, yes, there's going to be differences in the cyst lining epithelium compared to what it was when it started out. But we don't know whether those are chicken or egg. I think that's been the million-dollar question in the field. What are the changes that happen early that start the cyst off, that get it on its career to becoming a cyst? And those could be changes in the permeability of the cells or the membranes. They could be changes in the strength of that cell layer. But they could also be something totally different, right? They could be something at a signaling level, or the way the cells are dividing, the growth of the cells, the metabolism of the cells, right? So distinguishing like what is that process that goes from tube to cyst, that's still very complex and hard to understand.
I guess the difficult thing is that these proteins that are mutated or that are, as you explained to me, that are less active or less present, basically. So, for example, with somebody who has a PKD1 mutation, they would have maybe just half as many PC1 proteins as someone else that doesn't have PKD would have, right? And the difficult thing is that we probably don't even know all the functions that this protein even fulfills, right? Because it's not just one. We think of tools in our environment as having one job. But these proteins, they do multiple things, right? So just the two things that I always talk about is just it's like this Velcro sort of activity between the cells for the PC proteins, and then sort of regulating the calcium level. But I'm sure there's probably a lot more than just those, right?
Well, there's two proteins, as you know. There's PKD1 and PKD2.
Yeah.
One of them looks like kind of a receptor, so it looks like a signaling protein, and the other one looks like a channel, maybe a calcium channel. They are probably doing multiple things. They're in multiple places of the cell. For example, we know that polycystin 1, PC1, tends to be directed to the primary cilium, which is this little antenna that sticks out of the cell. PC2 will go there too, but its home is really more in the endoplasmic reticulum of the cell, which is a totally different spot. So how this all works, that's a beautiful puzzle that nature has set forth before us.
So maybe one idea for the center in the future, which we have to talk about in a second, would be to sort of petition to rename the PC1 and PC2 proteins to something that describes their function, at least as far as we know it. Because now we're describing it by what happens if you don't have it, right? So why not give it a name that gives patients a lot better chance of understanding what it's actually doing? But that's maybe a discussion for another day.
Well, we could talk about the center, I think. That would be a great thing to discuss.
And I will ask you about that, but one final question on this. A lot of listeners, of course, are very familiar with the glucose connection. And in your presentation, you also had a part on the glucose transport into the organoids, which I think is relevant to listeners also because, of course, we now have at least one medication that sort of can influence that.
Yeah, this was really interesting, when we started looking at the cysts and we realized that this wasn't a secretory mechanism that was driving their expansion. So we said, what is it? And it turned out that it was really an absorptive mechanism. They were pulling fluid in from outside the cyst, and that's how they were growing. And the way they were pulling it in, the reason they were pulling it is because they were pulling in glucose. And they were essentially accumulating glucose inside the cyst, and water was essentially coming in to equilibrate between the concentrations on the outside and the inside of the cyst.
So what you're saying is it pulled in more glucose, which sort of increased glucose concentration inside the cyst, and then it had to pull in more water so that is balanced out?
So this is the way tissue compartments often work. They don't like having a huge disequilibrium between the outside and the inside because that will create an imbalance and makes them uncomfortable. So instead, we've developed all these channels that can rapidly move water through cells, for instance, to equilibrate when there's a disequilibrium between one compartment and another compartment separated by a layer of cells. So that's right. The water follows, and water will also follow not just sugars, but also ions and larger molecules.
So in the case of PKD, you were talking about glucose specifically, and the transporter that pulls it in is the SGLT transporter?
Right.
Maybe you can sort of hint at what we can influence today with medications or even diet.
Well, the SGLT2 transporter in particular is strongly expressed in the kidneys. What's happened over the years is that this class of drugs, the Flozin drugs, have emerged that can block those transporters. And these drugs turn out to be really helpful for patients with heart disease or kidney disease in a broad spectrum sort of way. So they're now essentially one of the major pillars of drugs used to treat chronic kidney disease. They're really popular. It's been a success story that's only really emerged over the last decade or so. After a long drought where there really wasn't a lot of new drugs for folks with kidney disease. So now the question becomes, what would they do in the context of polycystic kidney disease? Or what are other types of interventions? Maybe there are natural interventions that you could also pursue to lower exposure to glucose, for instance. So I think these are fair questions. They are more on the clinical side than where we stay. We live on our little organoid world, and we are trying to use that as a platform to make these types of insights. And then it's handed off essentially to the clinical folks. And I know there are clinical trials ongoing for some of these SGLT2 inhibitors, for instance. So I'm eager to see how those turn out.
Yeah. And in a general sense, we can probably extrapolate from your results and then from also clinical experience that more glucose is probably bad. Having more glucose in the blood, being diabetic, there's one mechanism how that can increase cyst growth, right?
Yeah, we're all sort of addicted to glucose. Our cells are all addicted to glucose. And I'm not knowledgeable enough to really judge how that all works biologically. I think all metabolism is complicated, right? And you need energy from somewhere. But certainly, I think I would be on fairly firm footing to say that, the high sugar diets that have been very popular in the Western world are probably not great for our bodies and probably not great for PKD either.
So shifting gears a little bit, you recently applied for a very interesting grant and you got it. And now you opened the first, or is it the first PKD center or is it just your first?
Well, it's our first. It's not the first. The program's actually existed for about 30 years in one form or another. But this is a very prestigious program. It is run by the National Institutes of Health, the United States National Institutes of Health, and it's dedicated to polycystic kidney disease. It is a consortium of different centers which are based at different universities. Each of these centers endeavors to generate tools and data that the community can use for PKD research. So resources. And that's why it's called the PKD RRC, or Research Resource Consortium. And it's been around for a long time. And this is actually how many of the tools, many of the methods, many of the reagents, cells, animals, et cetera, in the field are distributed. So for us, we're excited about it. We're new to it. We're sort of the new kids on the block at the PKD RRC. And our center is dedicated to in vitro models, so things that are outside of the body, non-animal, in a dish systems, with our organoids being the flagship.
You sort of filled me on that before. There's going to be a list of things that might be interesting to test, to try on the organoids and maybe on other models that you guys are using. And there's going to be some way for the public to interact even, and have a little bit of an input on what they would like to see studied in PKD next. Is that about right?
Exactly. Yeah. So one of the things we've realized over the years is that it can be difficult for a group to just adopt the whole dark art of creating organoids. So we've established a service platform where people can send in their molecule of interest, their treatments, and we will do all the grunt work of making the organoids and testing out those molecules on the organoids for them and send them back the data. And for this PKD RRC, what our center is going to do is provide this as a annual community screening activity. So we're going to solicit entries, people to submit their molecules to us, and then we will run them all in a screen and provide back the data. And it will require interest, and it will require also some selection because we won't have the resources to do every single compound and obviously every single concentration of those. So it's more geared towards pilot studies. But yeah, it's a great way if you have an idea and you have a molecule in hand. Definitely that will be hopefully up on the website very soon. The PKD RRC has a central website that you can visit and see all the offerings. And more generally, we're really interested in hearing from the larger community, including patients. We are charged with being innovative. So we want some off-the-wall ideas. And there is a special group within our consortium that is trying to solicit these from the wider community. So ideas, expertise, things that haven't been tried in PKD. I mean, if you think about it, sort of like the organoids have been kind of a disruptive technology. Yeah. There are other disruptive technologies out there just waiting to be applied, to be born for this disorder.
So basically it's crowdsourcing some new things to test, and this could be a medication that hasn't been tested yet, could be some kind of supplement, it could be some kind of new molecule, some kind of peptide, and basically if there's a solid reason to test it, then people can sort of apply to have that included in the vote.
Yeah, and you can sign up. Every month we have a research seminar which is open to the public. You can sign up and check out the PKD RRC website. You can also check out my own website, which has some information about our center particularly.
And we'll have that in the description.
Yeah, and get involved if you're interested in this type of work. And we'd love to hear from you. Can't guarantee that your wacky idea is going to actually become an experiment in the near future. Those things are always a little tricky to guarantee, but just plant the seeds. You never know.
Well, some of them will.
Some of them will.
Yes.
Some of them will. And we want to hear from people who have PKD. We want to connect. We want to build community. That's also a big part of it. A lot of it is education. For anyone who's a young scientist in this field, in an educational program, my center is going to have a meeting. We're going to have an annual meeting, essentially a summer enrichment program, where we talk about organoids and how to use them. And it's going to be a 2-day meeting, which we're accepting applications right now until May 1st. So if you're interested in learning and coming to Seattle, there's an opportunity to do that as well.
That's amazing. I think people will actually love this. And especially in this community, there might be a lot of people that are inclined to share some of their ideas with the center. And I hope we can facilitate that.
That'd be great.
All right, man. I think we already went a little bit over time. Is there any last things that people should check out? So I'm going to definitely have the links in the description.
I think those are the major ones. If anyone wants to donate to the cause, so to speak, there's also a donation button on our webpage. We always welcome those types of things, and every dollar goes to research. So
that's great.
And if you're a big company that wants to do research in this area, you might want to check out our contract research organization, Plerexa, which we've spun out specifically to do larger projects, which are geared towards drug development. And we have an arm associated with PKD where we can do these types of services at scale.
All right, cool. Well, it's been a pleasure to have you. And I think we covered the basics. Maybe we'll do a second one in the near future. I do have some other questions that we might be able to cover then, but maybe you will have some new insights until then as well. I invite people to check out the website. I have it in the description. Thanks for coming.
You're very welcome. Thanks for having me.